Okay, so today we're going to go over the end of chapter 4. So this is part 2 of chapter 4. I went over part 1 before. And we're going to talk about protein production. And the terms that you're going to have to know, and you're going to have to know what these things are transcription, translation, and then secretion is when the protein that we're going to make actually moves its way out of the cell. So first of all, to talk about gene expression, if you don't know what a gene is just part of, I know we've all heard the term gene, but specifically, if this is DNA, a gene is just a part of that DNA, and that's going to code for a protein, or at least part of a protein. Okay, so when we have a gene mutation or something like that, we're not so worried about what it's doing to the DNA. The DNA is just going to go on just fine. But what happens is that protein, where it may be supposed to be shaped like that, if you have a mutation in the gene, which causes a different amino acid to be expressed right there, it may go the other direction or something. So it may not fold properly in this area, and then you have a protein that is just not going to work, and that's a mutation. That would be a bad mutation, and if you need that for something like vision or some other process, well, that protein isn't going to work. And so unless you have, since you do have two copies, you have one copy of genes from your mom and one from your dad. Well, if this one's mutated and this one's okay, you might be all right. You might still be able to make it because this one will make the good copy. If, however, both are mutated, well, then you don't have any good copies, and so that's going to be something you express. Okay, so there's a lot of I just summed up a lot of genetics just talking about this, but that's essentially what we're doing. We're trying to read the DNA to make a protein, and usually if you're walking around, you don't have any mutations that are that bad. Okay, so everybody has some mutations. Everybody's a little bit different. Their DNA sequence is a little different. So their protein might be, instead of shaped like this, it might be, shaped like that. But as long as it's functional, you're okay. So so that's where it comes in, and that's a lot of our genetic differences. They're very subtle. Okay, so all nucleated cells except germ cells have the full complement of DNA. So that means in every single cell, in the nucleus of every cell, you have 23 pairs of chromosomes. I can't draw all 23 there, obviously, but you have 23 pairs. Now, if you are a girl, then one of those pairs will have you'll have two X chromosomes. If you're a boy, you'll have an X and a Y. The rest, the other 22 pair, are all pretty much the same. The only difference is going to be the sequence of these bases in the actual DNA. Okay, so this is the only difference. Is if you're a girl, you have two X. If you're a boy, you have an X and a Y. Now, most of the time in a girl, one of those X's is turned off, okay, because we only need one X chromosome to get those proteins produced correctly. Okay, and we'll come back when we talk about reproduction. We'll come back and see what this Y chromosome is doing. Okay, so first of all, let's get reoriented with the cell. This is the actual cell, and inside the cell, we know that there is a nucleus, and we already talked about chromatin, which these are the unfolded chromosomes that exist inside the cell. Okay, so like I said, 23 pairs, they're all unfolded in a functioning cell, and they're being transcribed. Okay, and so they're going on to make proteins. Whichever proteins are needed for that particular cell, that's what's turned on. Okay, we see this thing called a nucleolus. I'll say more about that, but that's where something called ribosomes are made. Okay, so it's a darker area where ribosomes are made, and and then we make a copy of an mRNA that comes out through the nuclear pore. So we kind of went over that before, but now we want to get into more specifically exactly what's going on. Okay, so if we look at, I've been saying that the chromosomes are all unwound. And that's true. A lot of times we're used to seeing this right here. That's a chromosome that's getting ready to divide. Okay, everything has been packed. It's not functioning. You can't get the RNA polymerase to come in there, which is the protein that actually codes for or transcribes that to make the protein. None of that can get in there, so it's all packed up. Usually, how we see it is we see it like this. It's unwound, and there are these little histone proteins. That's what those are called, that wraps it around, and it's a very organized type of wrapping type of uh, structure. So then the proteins that actually have to transcribe that can fit on there. Okay, they can't do that in this case. It's kind of like this would be kind of like if you wind up a string or something and it all clumps together. That's what this looks like. Um, Okay, so, and then we just mentioned chromatin. When a cell is active, DNA is complexed with proteins, which are histones, in a cloud of DNA, and that's what you see here. Okay. All right. So, what are we really after? So, I have here. Let's make a protein. A protein that we've all heard of and that we've talked about is insulin. It's a pretty small protein overall, but you can see that the protein is a string of amino acids. Okay, and I mentioned before there are 20 amino acids to choose from. So you can kind of go through here. You can see threonine, lysine, proline, threonine, tyrosine, phenylalanine, glycine. And so these are all just different amino acids, and they have to be in that order because see, for instance, right here you have two cysteine amino acids, and those have sulfur groups on them, and they have to join up because they have to make the protein in this shape. Okay, the protein has to be in that shape, and so these cysteine residues, that's what we call that. These cysteine amino acids have to come into contact with each other to make it in that shape. Now, if you had a mutation where that cysteine was something else, like another valine or something, then you wouldn't have this, and that protein would unfold, and it wouldn't be any use. Okay, so now we can see here. Here's another example of a protein. This is it unfolded or denatured. And then this is the protein when it's folded. And so shape matters, okay? So it really matters how this folding takes place because a lot of times it's the folding. You may have to have this kind of angle with these particular residues right there to make something function, okay? So this shape is very, very important. Okay, so since we're talking about insulin, and we'll try to focus on insulin as our example, it's a small protein. It's made in the pancreas. It's made in beta cells of the pancreas. Um, and the reason we know so much about insulin is because of diabetes. And in type 1 diabetes, these beta cells are attacked by the body's own immune system, and it takes them out, and so you're not able to make insulin. Now, in type 2 diabetes, the insulin is still being made, so we don't have that issue. But we do have the insulin that's being made doesn't function. Okay, so it's not because of a mutation, it's because whatever it's supposed to bind to, we don't actually know why. We know it's not mutated, but for some reason, when it tries to act on receptors on the cell, it doesn't do what it's supposed to do, which is to bring glucose in. Okay, it's not bringing the glucose in like it's supposed to, so the glucose is remaining in the bloodstream. Okay, so if we want to move through this step by step, okay? So if we have our cell here and our nucleus with our DNA in it, okay? So we're just going to show a little short piece of DNA. We want to take that signal. The DNA has to stay inside the nucleus. The DNA doesn't leave the nucleus. It's protected. You know, there are things there's melanin around here that's protecting it from sunlight. It's guarded in here because any kind of mutation can cause bad things to happen. Okay, so what happens is that it stays in there and we make a copy, okay? Now, we don't make a copy of the DNA. We only find this section, a gene, then we say, okay, we want to code for the protein that's on this part of the DNA. Okay, so there's usually something called a promoter region, and there's a protein called RNA polymerase, and it will make DNA is double-stranded, and it will make a single-stranded messenger, that's what M stands for, messenger RNA. Okay. So we've got a couple of terms here. First of all, transcription, and you're going to have to know what transcription is. It's going to be a test question. And the easy answer is to say, well, transcription is where messenger RNA is made. A messenger RNA copy is made. It helps a lot to kind of understand what we're talking about. RNA is very similar to DNA. DNA is double-stranded, okay? That's some of the redundancy, that's how it fixes itself, it's double-stranded, okay? But we've also talked about these bases, okay? These are nucleotide bases. And there's some faithfulness to these bases that cytosine always matches up with guanine, okay? So this yellow always matches up with the green, okay? So that's what we see here, yellow matching green. And if you look throughout, yellow matches green. The colors are kind of washed out on my monitor. And you can see here, orange always matches blue, okay? That's, well, I guess I should say the red always matches blue, okay? Red matches blue, because this is what we see in DNA, these 4 bases, okay? Because I know, it's funny because I'm saying 4 bases, and yet there are 6 here, okay? Actually, there are 5 because adenine's repeated, and I'll talk about what that is. Because if you're just talking about DNA, you have these top 4, okay? The 4 that I outlined here. That's DNA. The only difference, and this can be confusing as you want to make it, the only difference is that RNA doesn't have thymine. Instead, it has uracil. They're very similar. They look very similar. I don't know why that it's that way, but whereas you always have thymine bonded to adenine in DNA, in RNA it's uracil with adenine. So here we can see in the DNA, the red goes with the blue. Okay, so this red right here goes with the blue. And we can kind of see that one right there. We can see the red and the blue matching up together. However, in the RNA, when the RNA is made, then it's the orange that matches up with the blue. Okay, so that's what we see up here. We see orange matching up with blue. Okay, so that's the only difference. It still has the sequence. The sequence is the same, and that's what's important, is because we have to keep this sequence together, because that sequence is ultimately going to code the protein. Okay, so we have to keep that sequence. So that's basically all that's happening. Is here's your DNA. It's just sitting there. You say, hey, I need to turn this gene on. For whatever cell you have and whatever protein you need to make in that particular cell, you say, oh, you know, the code for that protein is in the nucleus. I need that, but the DNA can't leave, so I'm going to make an RNA copy of it. And the RNA is single-stranded, and that's good enough for what it has to do. And it makes a copy, and it keeps that sequence of these individual bases, cytosine, guanine, thymine, adenine. 4 different letters to the DNA alphabet. It's actually pretty impressive that we can come up with all these different proteins and life in general from 4 different bases. Okay, so so that's what transcription is. We're making a copy. We make an RNA copy. And then that RNA, after it's made, transcription ends at a terminator sequence. After that RNA is made, it moves out of the nucleus, and then it's down here. So this part here is transcription. Okay? That happens, so we have to think about possible questions. What is transcription? That's making an RNA copy from the DNA. Where does it happen? It happens in the nucleus. Okay? And then that RNA can move out. And that's where it meets something called ribosomes. Now, we mentioned ribosomes before. Ribosomes are just proteins, but they're kind of special proteins. They're proteins that have a little bit of DNA in them. Okay, that's why they're made in the nucleolus. Okay, they have a little bit of, actually they have a little bit of RNA in them. Okay, and these things, because they have that RNA in them, and they're proteins, they're able to bind to the messenger RNA. Okay, so here's that messenger RNA. Ribosomes are made to stick to that. Okay, I like to call ribosomes translators. And guess what step is coming up next? These are translators. So, and if you guess that the next step that's coming up is translation, then you're correct. Remember, here was our nucleus up here. The RNA left, and now it's finding a ribosome, or a ribosome is finding it. They're finding each other. Okay, so ribosomes can either be freely floating around, okay, or, oops, that's a horrible ER, or they can be attached to the endoplasmic reticulum. Okay? So the RNAs or the ribosomes might be here, and again, the messenger RNA is going to find it there, or they could just be floating around, and the RNA will find it there. Okay? And then it will be translated. Okay, okay, so ribosomes. So here, if we look back, this is showing how the ribosomes are being made. Ribosomes are being made in the nucleolus, and this is what they look like. Okay, I drew a simplified version of it, and this is also a simplified version of it. But they're proteins. They're proteins with a little bit of DNA in them, and so they see this messenger RNA and they clasp onto it. Okay, they grab onto it, and then they can start the process of translating. So ribosomes get pieces of their own RNA to help them in the translation process, which that occurs in the nucleolus. So when you look back and we've seen that word, we've seen nucleolus, and I haven't said too much about it because I knew I'd be talking about it now. But that's that dense part of the nucleus, and that's what happens in there. That's where ribosomes are produced. Okay, so now remember we made a copy of the DNA, and now we have this single-stranded RNA. Okay, so remember we were in the cell, the nucleus. We made a copy of the RNA, and then the RNA moved out. Okay, so we have this single-stranded RNA. Now, it's going to have, of the 4 bases, so remember in DNA, it was G, C, T, and A. In RNA, it's G, C, U, and A. And these guys will always go together, and these guys will always go together. So GC doesn't change from DNA to RNA. It's just this T that changes for some reason. Okay, so we're interested right now in RNA because we're getting ready to translate this messenger RNA molecule that we just made. So if these guys can come together in several different combinations, so we can have, let's see, we might have G, C, A, U, U, C, G, U. Okay, so we might have this kind of a sequence together of these different bases. And so you say, well, how can we have 20 different choices for amino acids? These are a list of the amino acids. So we might say, well, gosh, 4 letters to an alphabet. How can we get anything done with that? But if we look at this, if we see a GCA, these 3 right here, GCA, then we know that this is going to be, if we look over here, and we look, and let's see, GCA right here. Means GCA is going to code for an amino acid called alanine. So that means that we have alanine. Okay? And that's what this is. And this is going to be something called the T RNA. Okay? Okay, and UUC is the next one. So if we go up here to the U's, we can see here UUC codes for phenylalanine. So that means this next one is going to be phenylalanine, and so on. Okay, so we can kind of make up whatever we want to make up. But you could translate that, and you could figure out the amino acid sequence of the protein, because this protein is going to be put together. All of these amino acids are going to be put together to make the protein, and we could figure out exactly what this is. Okay, so GUC valine. Okay, so valine is the next one. So then we have these identified. And it just goes on and on and on like that. And then we have a protein at the very end. Okay. So whatever this is reading, the anticodon is going to be the opposite. So G is going to go with C, C is going to go with G, A is going to go with U, U is going to go with A, and so on and so on and so on. Okay, so that's what we have to do. We have to read this messenger RNA molecule now. Okay, so that process is called translation. And that's exactly what happens. Now, I mentioned earlier the transfer, the tRNA. Okay, so tRNA. Okay, so remember we've already transcribed our messenger RNA. Remember that happened in the nucleus. Now we need to translate it, which means that a ribosome will stick to this, will bind to it, and then it's going to allow something called transfer RNA. I know, so many terms. Transfer RNA to come down and read these three things that we call a codon. Okay, and it's going to have the anticodon for that. So it's going to have those matching letters. I can't see this very well, so I don't know how well you can see it, but it's showing AUG, and then up here on the anticodon for the transfer RNA is UAC. Okay, so that matches. Okay, so that's going to drop off something called methionine. Okay, so that's the first amino acid, and that's always the first amino acid. We actually call that a start codon. Okay, so that's what we're doing, and so now we have to, all this ribosome has to do is just sort of move along that RNA from one to the next, to the next, to the next, and it just keeps adding amino acids. Okay, so this shows it nicely. It shows it starting here, perhaps with that methionine, and then it just adds and adds and adds and adds. So when you have something that's high in sugar and you say, gosh, you know, we need to make more insulin, then your gene will get turned on in the nucleus of these beta cells, and it will transcribe them to make the mRNA. The mRNA will come out, ribosomes will attach to it, transfer RNAs will then start coming down and dropping off amino acids, and then you have your protein. Okay, so that's what this is, your protein, your functional protein. Okay, so that's our goal. Okay, so if we look so far, what we have, the overview, so you don't have to picture it in your head, we had transcription that happened up here. Okay, so here's the DNA. Okay, so this thing right here is the DNA. And then we made a messenger RNA strand, and that process was called transcription. That messenger RNA moved out. I'll follow their arrow. And then a ribosome attached to it. So now we know what a ribosome is. Attached to that messenger RNA, and then the transfer RNAs, or the tRNAs, moved down. So tRNA move down, and each tRNA, and these guys are just floating all over the place. The tRNAs are floating around just waiting for their chance. Okay, so I mean, this guy was just floating around carrying its little arginine all over the place, just waiting for some space to open up. It's very full of these guys. Want some space to open up that it says, ah, you know, that's me. I match now. Okay, and that's what happened here. Here's our arginine. It says, oh, that's my codon. Okay, I have the anticodon for that. So GCU. So I'm going to come down here and I'm going to stick there. And then when you stick in that ribosome, it attaches to the next one, and so on and so on, and it moves right along. And there are videos showing this take place. And, you know, it seems like a really complicated thing, and it is, but to scale, it's moving along like this fast. I mean, it's just all the way down that messenger RNA. Okay, and then out of it, when it's finally done, out pops a protein, and that protein can then go on to be modified, and so it can actually work. Okay, so if we understand translation, or maybe did I skip? Oh, this step is called translation. Okay, that's why I call ribosomes translators, and if you remember that, then you'll know that it's taking place. So the question is, where does translation take place? And the answer is that it can happen in the endoplasmic reticulum. Translation can also just take place out in the cytosol. So our question is, where do we want the protein to be functional? If the protein just needs to be floating around, so this is I'll draw our nucleus up here. That's our nucleus. If the protein just needs to be floating around in the cytosol, maybe it's performing some function. Remember, glycolysis happens out in the cytosol. So those proteins would just be floating around out in the cytosol. However, some need to be membrane bound or they need to be excreted. So those have to take place on the endoplasmic reticulum. Okay? And guess which one it is? Is it going to be the rough endoplasmic reticulum that does this translating or the smooth? It's going to be the rough by definition, because the rough ER is covered with these down here. You can see it's covered with these ribosomes. Okay, and these ribosomes aren't just stuck to it for no reason at all. These ribosomes are translating proteins. So you can kind of see that here. Kind of see these guys translating proteins. This blue part right here, that's the messenger RNA. And this right here it's labeled here as polypeptide, but that's going to be the developing protein. Okay, so that's what we're making. That's what we're doing. And so that's one of the things that can happen here. And like I said, there are ribosomes out here that are translating, out here in the cytosol, that are translating proteins that will then function in the cytosol. Okay, so it can happen in both places. Don't forget that. Okay, so if we think about, if we kind of zoom in and we say, okay, ribosomes are translating, and we zoom in and we see, okay, well, this guy is sort of moving into, little by little, getting larger and larger, moving into the endoplasmic reticulum. Okay, then we can say, well, we know that maybe this thing isn't quite done yet. So one of the things that's going to happen here is maybe some of this is going to be cleaved off. Maybe it's going to be modified a little bit after it moves in, and we'll talk about some of those, a few more of those modifications here in a minute. But to start with, let's get a big picture, and let's say, okay, let's say that the protein that we want to make is insulin. Okay, so that's the example we started out with. So the protein we want to make is insulin. Well, insulin isn't going to be a protein that's just going to be out here in the cytosol. Insulin is something that we want to secrete. Okay, so we want insulin to move out. Okay, so that means that, and that explains why it went to the endoplasmic reticulum. And if you look at this picture down here in the corner, then here's the protein being made. It's packaged into these little vesicles. These vesicles move into the Golgi, which is going to modify it, cut some things a little bit more, and then those are going to be, it's going to be packaged into these secretory vesicles, and these, in this case, the secretory vesicle moves towards the membrane, the cell membrane, and it is excreted as functional insulin. Okay, so that's how these things work together. So that, hopefully, by remembering, your memory works better if you have a process. If you remember this whole process all the way through, you can kind of remember what these are doing because they're actually performing a function. So if we look at the Golgi, that's what's happening. It moves into the Golgi, and then the Golgi moves down here to the membrane, and then it opens up and it releases that, in this case, insulin, but it can be mucus molecules, it can be lots of different things. Okay, all right. So the question, what does modification mean? That's something that's always kind of bothered me. It bothered me when I took biology for the first time. They kept saying, oh, modification happens in the ER, and protein modification happens in the Golgi. But they didn't ever tell us what modification was. So I'm going to try to tell you what that means, modification. And if we look at insulin, the functional insulin protein, I don't know if you can see this very well down here, but the functional insulin protein actually looks like this right here. Okay, it's actually a couple of lines of amino acids together, and they're attached by disulfide bonds. So that means that this is not a full protein. Okay, it's not around like that. It's disconnected. So that means at some point along its way, it had to be cut. So it may have been formed like this, but then this had to be chopped off. This had to be chopped off. Okay, that's a modification. Okay, it's inactive before that. We call that pre-pro or pro-insulin because it's not ready to be made yet. So folding. Okay, it has to be folded properly. So as it's first coming out, it's not folded properly. Okay, it looks like this. Okay, it's just kind of this big string of goo. And so it has to get this proper folding. That's a modification. So this is going to happen in the endoplasmic reticulum in this case. And there's something else to look at. Okay. So this is the full protein. It's not the active protein because remember we have to cut it here and maybe cut it here to make it active. But there's something else it has. It has this attachment sequence. This is how it knows that it's supposed to be in the ER. So it's got this region. So that has to be cut off as well. Okay, so lots of things have to change. It doesn't just come right out of the ribosome ready to go and ready to be active. It has to be changed, which means that it has to be moved around to the ER. There have to be enzymes there that can cut it in the right place, and there has to be enzymes in the Golgi that cut it in just the right place. So that stuff is always going on. Okay, folding, cutting off the signal peptide, that's what I was referring to here, is the signal peptide. This is saying, hey, I need to go to the ER. That sounds the emergency room, but that's what it has to do. It has to go to the endoplasmic reticulum and it has to attach there. And so after it's made, it gets cut. Okay, so that's one of the things. And then cutting off extra amino acids. I have here something called the C peptide because after you cut this region off, okay, to make the complete protein, this pink area right here, that just goes floating away, and we call that a C-peptide. And that's actually a way to see if somebody's making insulin. We can actually measure to see how much of this C-peptide is being made to find out if someone is actively producing insulin. If you see an increase, that means they're actively making insulin because you can see that C-peptide right there. Okay? Just a little FYI for you. So the enzymes for these modifications are only in the places where they are supposed to occur. So that means that everything has to function right. The beta cells have to be right. Inside the beta cells, inside the Golgi apparatus and the ER of the beta cells are enzymes that will cut this in just the right place. Okay, they may not be expressed in other cells, but they're definitely there. Okay. And then it moves out. Okay, so hopefully we kind of get that idea that then the protein that in this case is insulin will be packaged into the secretory vesicle and it will move out. Okay, so we have examples, other examples, digestive enzymes by the pancreas, which would be insulin, mucus by salivary glands, secretion of milk by mammary glands. Same thing. This is what's going on. We're producing proteins when the mother produces proteins and when she produces certain antibodies. These things are being made and they're being ejected out with the milk. They're being expressed with the milk. Okay, so cells that are responsible for a lot of excretion would probably have a pretty extensive, I mentioned this earlier, and that's going to be they're going to have an extensive Golgi apparatus. Okay, so this is just an overview of everything we just talked about. Another one, just so in case you want a different way of looking at it, but transcription happens here. mRNA is made, attachment of the mRNA to the ribosomes or the ribosomes to the RNA, translation, which involves these tRNAs to put the amino acids in order, and so you have a completed, this is a very short peptide, but a completed protein. That's ultimately what you want. And usually proteins are thousands and thousands of amino acids long. Okay, they're usually not as small as the insulin that we just saw. Okay. All right, that's it.